So if we're releasing the hormones from our pancreatic islet, then that's our glucagon and insulin which affect the blood glucose level, whereas if we're secreting our enzymes from the Acini cells, then these are going to exit the pancreatic duct into the duodenum of the small intestine and assist us in breaking down our food that has reached that point in the gastrointestinal tract. So we're actually able to absorb the breakdown products of the protein, carbohydrate, and also the lipid. So we've got the hormones and enzymes going to different locations because they're needed for different roles, and they're going to essentially where they're needed. So we've talked a lot about the regulation of blood glucose levels, but we haven't really worked out how this can actually be controlled. So we said that the glucagon is going to increase the blood glucose level, and we said that insulin is going to decrease the blood glucose level. But what's to stop the insulin from decreasing the blood glucose level too much? Equally, what's to stop the glucagon increasing the blood glucose level too much? So what actually stops this is the process of negative feedback. So when the blood glucose level drops too low, this actually will inhibit insulin. And if the blood glucose level gets too high, this will actually inhibit our glucagon. So that's what we have on the left-hand side here. So to summarize that, we've got the points on the left-hand side here. So basically, if you have a low blood glucose level, that's going to stimulate the glucagon release, but equally, it won't allow insulin to be released because insulin acts to lower the blood glucose level, and we don't need it to be lower. So this provides negative feedback to insulin to basically stop it being released. And it's quite similar for if we have high blood glucose levels. At higher blood glucose levels, this stimulates the secretion of insulin, but equally, it stops or inhibits the release of glucagon because we don't need any more glucagon. The glucose level of the blood is already high, so we don't need to release any more glucagon to make it higher. So this is our mechanism of negative feedback. So basically, the negative feedback prevents the blood glucose level from going any higher by releasing more glucagon, and it prevents it from getting any lower by releasing more insulin if you're already in a state where the blood glucose level is low. So it allows us to stay in that nice sweet spot where if we have low blood glucose level, we can increase it a bit by releasing glucagon, whereas if we have a high blood glucose level, we can decrease it a bit by releasing insulin. We never go too high or too low because of this negative feedback mechanism. So what actually allows our glucose to come inside the cells? So we've talked a lot about how insulin is able to assist us in bringing glucose into the cell, but there is actually a mechanism for how this happens. So there's actually a reaction between insulin and what we call the insulin receptor on the cell surface. So that's what we can see in the red circle here. We've actually got the insulin receptor on the cell surface, and we've got insulin. So when insulin actually binds to the insulin receptor, it actually promotes the opening of the the glucose transporters. So we can see the glucose transporters are actually recruited to the surface of the cell here. So we can see the cell membrane represented in yellow here. So before insulin had bound, there's no none of these glucose transporters on the surface, but after the insulin receptor binds insulin, then we get the recruitment of these glucose transporters to the surface. So what this allows us to do is actually bring glucose inside the cell. However, after insulin levels begin to drop, what actually happens is, instead of keeping these glucose transporters on the surface, we actually start creating these vesicles, which allows the glucose transporters to come inside the cell, so they can no longer function to bring any more glucose inside the cell, because we don't have the signal to do this. The insulin has stopped signaling us to bring glucose into the cell. So we can actually then reuse these vesicles to bring the glucose transporters back to the cell surface in response to insulin again, which is telling us to bring the glucose inside the cell. So it's basically the process of the insulin binding to the insulin receptor, bringing the glucose transporter to the surface, which allows glucose to come inside the cell, and then in the absence of insulin, we take the glucose transporter away from the cell membrane until it's needed again. So why do we actually need the glucose transporter to bring glucose inside the cell? So this is because glucose on its own actually can't move through the cell membrane. We need a specific protein called the glucose transporter to facilitate this process. So how does insulin actually control when the glucose is brought inside the cell? So there's actually a receptor on the cell surface, the insulin receptor, that binds to insulin and brings the glucose transporter to the surface of the cell. And when it's at the surface of the cell, it can allow the glucose to come inside the cell, the glucose that is. What about when insulin stops activating this process? What happens then? Then the glucose transporter is actually taken away from the cell surface, so no more glucose can be brought inside the cell. So this slide links back to what we were talking about earlier with controlling enzymes by using covalent modifications of adding or removing a phosphate. So we can see on the left-hand side here that glucagon and adrenaline both activate a enzyme called protein kinase A, and it does this by activating cyclic AMP. So we activate directly the glucagon and adrenaline will activate the cyclic AMP, which then activates our protein kinase A, which will actually phosphorylate a certain set of enzymes. So these set of enzymes will actually promote glucose being made and or stopping glucose usage. So we're going to stop it being used for energy and for storage, and we're going to make more glucose. So both of these are allowing us overall to increase the blood glucose level. And we do this by upregulating or activating the enzymes that favor making glucose and deactivating the enzymes that will actually be involved in using glucose. So we want to stop using glucose and start making more of it, so we can take that glucose out of the cell and back out into the blood, so we can raise that blood glucose level. So how are we actually controlling these enzymes, whether they're going to be active or not? So it's through adding the phosphate via our protein kinase A. So the phosphate will activate the enzymes involved in processes that make glucose and deactivate enzymes involved in processes that actually use glucose. So insulin works in a similar way except insulin will actually activate a protein kinase which will actually dephosphorylate. So essentially what's happening is through cutting off the phosphate on particular enzymes, we're actually able to promote the reactions and metabolic processes that allow us to actually use glucose. So these are to make ATP or to store the glucose. So essentially what we're doing using our protein phosphatases is the reverse of what we were doing with our protein kinase. So the protein kinase is adding phosphate, and that was actually deactivating our glucose utilizing processes in terms of making ATP from glucose and storing it. So instead what we use is our phosphatase, and that will actually remove the phosphate. And by removing this phosphate, we're actually able to activate the metabolic enzymes responsible for allowing us to generate ATP from glucose and also to store it. The other effect of removing the phosphate is we're actually going to stop the processes that are making more glucose. So we're going to inhibit our gluconeogenesis and also the glycogenolysis, which would break down the glycogen. So this links quite intricately to what we were looking at right at the start when we introduced the hormone control of blood glucose level. So if you're a little bit unclear about what's going on here, I advise you to look back at that one because it links in quite well. Alright, so we can see here that the phosphorylation of the insulin receptor is actually able to speed up the uptake of glucose and amino acids into the cells. So we actually have specific receptors, a specific insulin receptor that will be binding insulin, and when we bind insulin to that receptor, let's say here's our insulin, that's actually promoting this phosphorylation, so the adding of the phosphate to that insulin receptor. So that's actually having this downstream process of speeding up the uptake of glucose and amino acids. So we've got actually our glucose receptor, which is our glucose transporter, GLUT4, which is allowing us to bring glucose into the cell. And we also have a specific amino acid receptor that's allowing us to bring amino acids inside the cell. So essentially what's happening here is the insulin binds to this receptor, and this allows it to become phosphorylated, and then that receptor actually signals to these other transporter proteins to say, hey, we need to bring more glucose into the cell and more amino acids. So it then brings those into the cell. And we can actually then do our downstream processes to use what we've brought into the cell. So we can do protein synthesis using those amino acids to build new proteins. And we can utilize our glucose to generate energy and also to store it as glycogen. And if there's an excess, we can actually start making our triacylglycerol, so we can store the excess glucose as glycogen, or we can actually turn it into fat. So the other thing that insulin actually does is it will inhibit the breakdown of adipose tissue and inhibit gluconeogenesis and also glycogenolysis. So essentially, insulin is stopping the breakdown of our carbohydrate and lipid stores and enhancing the utilization of these sources in terms of creating energy. And if we've got enough energy, then it will start storing them in the form of glycogen. If we're storing directly using the glucose directly to store, we can turn that into glycogen. Or if we've got enough glycogen, then we can start generating fat from the glucose. Alright, so what happens if someone has diabetes? So what happens to their insulin in terms of how they're producing it and how they're secreting it? So we have two different types of diabetes. We have type 1 diabetes, which is inherited. And we have type 2 diabetes, which is acquired. So if we have our inherited diabetes, this is where there is actually a problem with the beta cells that release insulin. So remember, our beta cells function to release insulin. So if there's some sort of damage to these beta cells, then the insulin is not released, at least not released to the levels that we need it to actually properly control the blood glucose level. Because remember, we need to release that insulin to bring the glucose inside the cell. The other problem we can have is that if we're releasing insulin so much due to a high carbohydrate diet, so if we're having a lot of sugar coming into the system, what will happen is the pancreas will keep releasing insulin, releasing more and more, and eventually what happens is your cells stop responding to insulin. So this actually again will lead to us stopping bringing glucose into the cell. So there's two problems we can have. We can either not have enough insulin to do the process, or we can have the cells actually not able to respond to the insulin properly to bring it inside the cell. So if it's inherited, then it's actually a problem with the release of insulin from the pancreas, whereas if it's acquired, you're developing some sort of resistance to your insulin because you've been releasing too much of it over a long period of time. So this is what happens when people have very high sugar diets over a long period of time. And that's why that predisposes people to type 2 diabetes. So what actually happens once you have diabetes? So essentially, the glucose uptake to your cells via the GLUT4 receptor, which we saw on the previous slide. So normally this is upregulated by insulin. However, the insulin sensing cells that will normally bring the glucose inside the cell no longer register that glucose is present in the blood because they don't sense that the insulin is there. So essentially they're not responding properly to the insulin to allow the glucose to be brought inside the cell. And the consequence of this is that the glucose is actually not brought inside the cell. So that actually is very dangerous, because if you can't bring glucose inside the cell, then essentially you can't really derive nutrition from carbohydrates. And even worse than this, it actually compromises other aspects of our metabolism that allow us to derive energy. So after we can't bring glucose into the cell, that's where the lipids become the major source for creating ATP. However, there's actually a block in this process as well due to the lack of carbohydrate digestion, which means ultimately we're not getting oxaloacetate. So in one of the lipid digestion lectures, we look into why there's a lack of this oxaloacetate. But it's essentially because you're not doing glycolysis, not creating pyruvate, which is used to create oxaloacetate. So without oxaloacetate, we can't actually do the citric acid cycle. So we can't actually create ATP from the aerobic respiration. So instead, we start doing ketogenesis, which is the use of ketone bodies to derive energy. So this comes with it a whole bunch of problems itself in terms of lowering the pH of the blood. If we go into a state of ketoacidosis, that can actually become really dangerous and lead to coma and death. So that's why for a diabetic individual, they really do need to seek treatment where they can start having the adequate supply of insulin and a type of insulin that they will actually be responding to properly to allow them to derive nutrition from both carbohydrates and lipids. So we've really looked at these processes in a lot of detail today. So ketochrome messages is basically that gluconeogenesis is the process that we use to increase the amount of glucose that we have, and we can take that back out into the blood to increase the blood glucose level. So remember, we start gluconeogenesis with a non-carbohydrate source of pyruvate, for example, breakdown of amino acids or from lactate. And then we looked extensively at the hormone control of metabolism, showing that if we have a low blood glucose level, then we need to release glucagon from the pancreas to increase the blood glucose, whereas if we have a high blood glucose level after a carbohydrate-rich meal, we release insulin, which lowers the blood glucose level by bringing the glucose inside the cells.